Good morning. I am delighted to welcome you this morning to our panel discussion entitled Reimagining Health Challenges in the Nonprofit Sector. I'm Tom Daniel, chair of TSRI's Board of Overseers and a former academic physician, entrepreneur, corporate R&D leader, and drug developer. But currently as an investor, an advocate, and enthusiast for innovation applied to the translation of science to medicine. It's a real pleasure to moderate this illustrious panel. I'd like to begin really with asking Mark to lay out for us what he considers maybe some of the dominant forces that are driving acceleration of translation into medical advances currently. In other words, Mark, what's working?
Thank you so much, Tom, for chairing this session. Thank you to Pete and colleagues for organizing this meeting. It's really a wonderful opportunity to tackle this issue together with Pete and Corey and you, Tom, as well. If I think about what's working, I always like to step back and think about what does it take to develop a new therapy or cure for a poorly treated disease. And at one level, conceptually, it's very simple. You need to identify the right target. You have to make the right drug against that target, and then you have to develop the drug in the right patient population. So it's easy to formulate the problem that way. Of course, each of those steps is extraordinarily difficult. But if you ask what's been working, why are we in a better place today than we were 20 years ago or even 10 years ago? I think there's been progress on all three fronts. In terms of identifying the right targets in disease, there's just a huge acceleration of knowledge of what goes wrong in disease that's been fueled by very powerful tools to study model organisms, tools to study cells, methods from human genetics, and also imaging and diagnostic modalities to use directly in human beings. So all of that is fueling a huge advance of the body in health, if you will, and what goes wrong in disease, helping us get closer to the right targets. There's also, in terms of developing the right drug, there's huge progress. The big difference today compared to 20 years ago is just the explosion of drug modalities. So beyond small molecules and antibodies that were already, well established then, we also now have various kinds of gene therapy, delivery of antisense, of mRNA, of DNA, different kinds of modality, viral delivery, and other kinds of delivery. We have cell therapies where cells replace other cells, and we also have cells as delivery vehicles themselves. So there's just a larger menu of drug modalities that can be used to tackle different diseases, different targets in different diseases. There's also, I think, a better understanding of what it takes to move from knowledge to drug. People have realized you really want to go for big effects. You want to really focus on increasing the potency and specificity of your drug and its bioavailability before you go into trials, something that was not attended to as much 25 years ago. So we know a lot of the don'ts and people avoid them. So I think all of that together has resulted in an acceleration of translation, I think your words, of science to medical advances. Another key element here is more at the institutional and organizational level. We have a more connected ecosystem with different parts of the ecosystem playing different roles. Universities really overweight in generating fundamental knowledge and the first steps of translation, small biotechs and disease foundations, helping take that knowledge and move it forward. And of course, larger biopharma companies moving things further along as well. So the many elements have progressed dramatically over the past 20 years. There are, of course, big obstacles and gaps. It's not, we aren't where we want to be, but I think it's important to recognize how far we've come in the past 20 years.
That's terrific. Corey, maybe you could give us your perspective on this, in particular, amplifying on roles that the public sector has played through initiatives like BRAIN, and maybe from your perspective in the Chan Zuckerberg initiative.
Again, if you want to think about what we're doing really well, I think a lot about the academic sector and supporting science and technology. And that my charge at the Chan Zuckerberg Initiative is to support basic science and technology that will lead to make it possible to cure, prevent, or manage all diseases by the end of the century. In other words, to take a really long-term view of what kinds of discovery would be necessary to move the field forward. And there are a lot of examples where where the academic sector has been the initiator of change, the innovator, the people who found the key initial discoveries. If we look, for example, at the field of cancer biology that was originally developed in academia and spent many years moving over into translation into drugs. If we look more recently at things like the amazing advances in computer vision, those started in academic theoretical neuroscience labs, Jeff Hinton's lab at Toronto, who developed the image analysis that then was taken up by Google. So we can look at these, we can say that these things are successful and they're good, and that's great. And I've been thinking about where, again, the gaps are, where we can make them more successful. And one is that I think in academia, we haven't quite embraced our role as innovators as opposed to educators in science. So if you look at academia, we have a very hierarchical, traditional system where the first class citizens are the professors, and then there are the trainees who are the postdocs and the students. And that's a great idea for education, but even there it has some shortcomings. And certainly in terms of actually driving science forward, there are some gaps in there. And thinking about where expertise comes into the system, where first class citizens are actually the people who know a lot about microscopy, or the people who are building new approaches to solve problems using whether it's computers or microfluidics. These people are sort of second class citizens in the academic environment. They're kind of working temporarily. They have to like recharge all their hours to different grants. And it's not consistent with what we really want to do, which is to have the science be at the highest quality. These are the people who innovate and educate and move fields forward, and embracing more of that, of the idea of not just educating the next generation of scientists, but educating them in an environment with truly great technologists is part of the goal of science as a whole. But I think it's something that, for example, in neuroscience, we've really taken to heart at the next generation of sort of funding and experiments.
Yeah, terrific. Pete, having watched the progress of Scripps over the last, I guess, 20 years, and in particular, marking the progress that was reflected in your talk about, a pipeline of translated science across the entire spectrum. Be really interested in your take on what the elements of greatest, sort of integrative value are at Scripps, and in particular, what parts of the infrastructure of an institute like Scripps are unique and powerful?
Yeah, so that's an interesting question, Tom. I would, kind of taking off of some of Corey's remarks, I think the thing that academic institutions in general bring to science, although we could do it better, as Corey said, is innovation. Okay, I think in the nonprofit world, risk taking for high reward outcomes is really nurtured from day one. I mean, people who are really successful are those who do new and highly creative things. And so I think that's probably the biggest asset that we can tap in terms of kind of addressing the gaps in the current in the current way drugs are developed. As you go from academic settings to biotech startups, a lot of the investors there are on 5-year timelines, which doesn't really, isn't totally compatible with risk-taking and innovation. And as you go to pharma, I don't want to offend anybody, but I think people become more afraid of failure than success, okay? So really figuring out how to capture the innovation and the brain power of the nonprofit sector and bring that more into the drug discovery process. And I think what we see at Scripps, and certainly probably at other institutions, is the attitude now of young faculty and graduate students and postdocs is not only make a great discovery, but they really want to play a role in accelerating the impact of that discovery on medicines that impact patients. So there's a huge opportunity here to begin to take that brain power and move it forward. And we've had great models like that in the past. Bell Labs was a great model when I was a grad student of basic physics, okay, bridging into applied physics. It created all kinds of devices that forever changed society. And can we begin to do that more and more in the life sciences is an interesting challenge, and how to do that and what do we need as infrastructure is a good question.
Mark, I know you've been at so many different interfaces in this sort of evolution in science and translation to products for patients. As you think about the elements that were most powerful in coalescing the points that Pete and Corey just made about the longer horizon line for basic science, and then the clarity of purpose around making a product out of that. Maybe you can speak to some of the key elements that were helpful in your spin out of Denali, your creation of other companies. I just love to hear your articulation of what are the key elements for the success.
Well, thanks, Tom. And maybe I can step back just a little bit, and I want to just continue with the train of thought that Corey and Pete initiated there about where the choke points are. Where is it that, as you go from developing knowledge about a disease, identifying a target, moving towards developing a drug and so on, where are the big hurdles? Because I think to further improve the system beyond what we have today, we have to understand what are the biggest bottlenecks, and then tackle them. And I think, Corey and Pete both touched on that. And maybe I can illustrate with how we've been thinking first when I was at Rockefeller, and then now at Stanford. And there are multiple bottlenecks. And for someone as a leader of an academic institution, it's not our role to try to solve every bottleneck. Some of them will be solved by others, by whether it's small companies, big companies, disease foundations, the government. But we thought that there are two areas where academia, and so this is more to illustrate the process of thinking through the bottlenecks and then saying, where can we with our institution make a difference? As having spent 8 years at Genentech, being on the receiving end of knowledge and trying to decide what we're going to focus on to make drugs, and then being back in academia on the other end of seeing our faculty make discoveries and push their discoveries towards industry. One of the bottlenecks, there are two bottlenecks that we've identified, that we've decided to focus on, first at Rockefeller and now at Stanford. The first is really going from target to what I'll call drug prototype, to use the engineering jargon. If it's a small molecule, it'll be going from target to early lead. And then, of course, we all know there's lead optimization to make the actual drug. But when you go to the drug prototype, here's the problem. The academic, the best academics will come up with a terrific idea around a particular target, which they believe, if they could make a drug against it, would ameliorate outcomes in cancer or asthma or Alzheimer's disease, whatever disease they're studying. And then they're at a loss about what to do next because they don't have the skill set to go from that target. They're very skilled at deploying cell biological, animal models, human genetics, all the different tools to identify that potential target. But then to go to a drug prototype to test it, they don't have the skill set. So they often start a random walk of talking to small companies, big companies, potential investors, trying to find someone to get interested in this. On the other side of the spectrum, you're in a company, I was at Genentech, looking at people pitching 1,000 ideas a day. Well, not 1,000 ideas a day, but 1,000 ideas a year. We evaluated over 1,000 opportunities a year just in oncology. And when you look at them, some of them seem compelling, but to really drill down, you'd have to recreate the whole infrastructure to study that particular target, those cells, those animals, et cetera, or you have to set up collaboration with the academic. So what you really would like to have if you're in the private sector is a drug prototype. So they can give you that, and you can test to see whether you believe in the hypothesis. And so we figured that a big bottleneck is that the academics are skilled at identifying candidate targets, but don't know how to make drug prototypes. Industry knows how to make drug prototypes, but is awash in targets and doesn't know which ones to focus on. There are two ways you could solve that problem. One would be to put a lot more biology in industry. That's not going to happen just because there's so much to do once you start to focus on the target. And the best companies do a lot of science, but you can only do so much. When I was at Genentech, we had 100 postdocs, the largest postdoc program in all of industry. At Rockefeller, I think we had 400 postdocs, and Rockefeller is a tiny organization, let alone how many you have at Scripps or we have at Stanford. We have 2,500 at Stanford. So it's just not going to happen. The alternative is to have drug prototype making in academia. And for that, because our brilliant scientists don't have the skill set to do that, we have to provide them with the infrastructure, as Pete said, and as Corey said. So what we did first at Rockefeller, and we did this in collaboration with Sloan Kettering and Weill Cornell, we've also done this at Stanford, is to create infrastructure so that academics can, on a competitive basis, access slots to go from target to drug prototype. They have all the cell assays, the animal assays, it's all set up. And skilled medicinal chemists and other scientists are there to help them do that. And they work together. We did this, we decided early on that it'd be better if we did it with a partner that really was very skilled as well. And in both cases, in fact, our initial collaborations have been with Takeda in New York and also at Stanford. And so this process of developing drug prototypes now, you've eliminated one of the big obstacles, which is the wall you hit when you have a promising target as an academic, but you can't go further. So that's an example of really thinking through what's the bottleneck and trying to create the conditions to go through. And by the way, Caliber, which Pete and his colleagues created, I think is exactly that. It's to go from a great idea around a target to making a drug prototype. And in some cases, you'll actually go ahead and make the drug substance too, but typically you would then partner with someone else to do it, right? But it could happen. But we all know, to go from an early lead in small molecules through lead optimization to a clinical candidate requires massive investment. And that's best done in the context of the private sector typically. The other point about this is, if you think about it, the current handoff is the academics get to the target, industry does the rest. But the natural scientific handoff is really at the level of drug prototype, because making the drug prototype enables the academic to test their theory. You can you can study the science further and see whether you were right potentially or you were wrong. At Genentech, our data were that of the targets that we initiated projects on, 60% of them would progress past the drug prototype stage. Many of them died because it turns out you had a prototype, but the theory was wrong, right? So for the scientists, it's important to know that. So it's a natural handoff. The other place, and I'll just keep this brief, is a big bottleneck right now remains identifying the right patients on which to test a particular drug. And the academic sector, we believe, can help also through the having, what I think everybody here on this call will know are, phase 0 hospitals. At Rockefeller, there's a phase 0 hospital. At Stanford, we're creating a phase 0 hospital network where scientists can actually test the biology of disease and test the effects of potential drug candidates with the aim of understanding the biology better and identifying patients who are responsive. Identifying that and identifying prognostic biomarkers and efficacy biomarkers and the like are so important to advancing drugs through the clinical trial process, as I think everybody knows here. And again, academia can help there. So 2 examples of bottlenecks where we can do our part to try to help blast through them. And for me, I think we have to look at the valley of death that we all talk about as a problem to be tackled with an engineering mentality. What are the bottlenecks and how do we solve each of them in turn, working together with different organizations, academia, the private sector, disease foundations, each doing their part, but with the same aim.
No, that's terrific. Pete, you I think were visionary about the way to create Caliber. It would be valuable to reiterate some of the concepts that you used to put it together. the logic behind bringing it into Scripps and the challenges that that creates going beyond.
Yeah, that's an interesting question, Tom. Caliber actually came out. I've been an academic all my life, but when I moved to Scripps, we actually had a unique opportunity that Novartis, the Novartis Research Foundation, actually over 10 years, gave us more than $1 billion to figure out first, how to apply genomics to medicine and the development of new medicines, but then to build technologies. So we built a lot of the high throughput technologies people see in drug discovery today, all the Stodley yellow robots and so forth. My chief engineer was from GM. And that was important. And then Novartis, because of the amount of money they were spending, forced us to actually use those technologies and tools to discover drugs. They posed, if this is so great, can you make a drug? Okay. And what was interesting is we didn't have anybody with drug discovery experience in the place. I mean, I remember going up to Nat Gray, who was really young at the time, and saying, "Nat, you think we can make a cancer drug?" Yeah, no problem, Pete. So we had this incredible opportunity with a lot of independence and a lot of tools to try and do this. And it turned out, with a little bit of luck and a lot of really talented people, being really quite successful, to the extent that Novartis internalized it. But that gave us the confidence. I mean, some of the drugs we put into the clinic at GNF came out of single postdoc research projects. Okay, so that gave us the confidence to try and do this at Caliber. And without that, I mean, the good news is we made a lot of mistakes, and we paved over those mistakes with Novartis's money. Okay. To their benefit and ours. You can't do that if you're trying to do this in the academic sector, especially, we're actually trying not only to make prototype molecules, we're actually, we've ourselves filed 3 to 4 INDs over the last year and a half, and probably 2 a year going forward. If you make a mistake in the academic sector, it's incredibly costly. Okay. And I think that's one of the challenges. How do you learn to do this with limited resources? And so if you are going to do this, you need a new way to get the resources to do this, because a lot of academic institutions are really unwilling to divert money from endowments to doing high risk drug discovery. And that's interesting because, as you think about a biotech startup, investors don't give money to biotech startups to put in an endowment to fund the startup. They invest in the startup to spend the money to create more medical value and financial value. Academic institutions don't do that in general. So there's also the resource question and how, especially if you're thinking about putting drugs in the clinic and failing, how do you fund all of that? And I would argue, as a somewhat related but separate topic, academic institutions have created huge value in medicines and financial value. I mean, look at the Bay Area, look at the Boston area, and now San Diego. But they benefit very little in the financial value that's created there. So I would argue we need to explore mechanisms where academic institutions who play a major role in creating this value capture a greater part of that value that they can then reinvest in the research enterprise, okay? And feel like they can spend the money to take more risk, especially if you're looking at drug discovery where failure is expensive. So I think that's also a major challenge in thinking about academic institutions doing high risk research, especially in the development of new medicines, it's costly, is where do those monies come from? And if you take them from the NIH, you're robbing Peter to pay Paul, okay? And where does that come from, and how do we capture greater value? And one of the themes of what we're doing by moving medicines further into the clinic is our people want not to make prototype molecules, but to actually make a molecule that they see being dosed in a patient. And, you know, the question, can they? I think we're starting to show they can. But B, can you create more value that way and capture that value and reintroduce it into the research enterprise to support these kinds of activities? So that's a huge challenge. And the beauty when we did GNF is Novartis paid all of that. The challenge with doing this in an academic center is how do you pay for it and have the monies you're willing to risk?
Corey, I'm very interested in two aspects that perhaps CZI may play in this sort of ecosystem context around the gaps. How do you balance the funding of basic versus applied initiatives, and how do you think about the manpower issues that are undoubtedly going to be a piece of making applied science work? You know, one of the fundamental differences, in my view, between physics and astrophysics and even large engineering projects that have changed the face of science over the last 20 years is that the difference between sort of the training of physicists as team scientists compared to training of biologists and chemists as individual scientists. And I think that there are efforts to make team science work in biology and in translation, but I'd really like your view of how that might best play and what CZI is thinking about.
Yes, thank you. I agree with what Mark and Pete were saying about scale. So scale is one of the things that you need to start to build into the process in order to make progress against disease. The other thing that I find missing is flexibility. So the ways that people work together, for example, in the COVID era, have shown that people who want to work on a problem together could very assemble into groups where one person would be the immunologist, one person would be the virologist, one person would be the structural biologist who would see how the antibody was interacting with the spike protein. And tremendous numbers of people suddenly found themselves working together who hadn't in the past, each bringing what they were good at for a short period of time to study a defined problem. When you have grant systems and these sort of inertial systems where you have to spend a year generating the preliminary data to spend a year writing a grant to start a project, that creates a real barrier for people moving forward. And it creates a huge barrier for people moving into fields that they don't know about. So trying to create not necessarily a top-down team science in the sense that, for example, someplace like CERN, which is absolutely limited as in physics by the fact that there can only be one facility physically of that scale, but rather flexible teams that allow people to bring expertise to a problem for a period of time and move on is something that we find exciting. Every time I talk to a physician, they tell me about some biological problem or puzzle that I don't know the answer to. I think, and as we learn more and more about medicine now through things like the large genomic studies, we suddenly realize how little we've understood about underlying mechanism and how much often basic scientists who know something about an underlying mechanism stand to benefit by working with physicians and on a problem that has a medical basis. So, Denali is a company that's working on Alzheimer's. The big shock in Alzheimer's that came from genetics about 5 years ago was that everyone was focusing on the neurons that were dying, and it looks like a lot of the risk resides in the brain's innate immune system, the microglia. It's like trying to figure out what's wrong by studying the victim of a car crash by studying someone who was run over by a car rather than the car that ran them over, right? You need to sort of see the whole picture. And having the flexibility to go in there mechanistically with some of the approaches we have now that combine, for example, a basic scientist who knows a lot about endocytic vesicles with a clinician in Parkinson's, where something about the endomembrane system is disrupted in many patients in a way that's fast and flexible, and again, doesn't require this huge sort of inertial buildup, is a place where I feel like there's a bottleneck that we can start to build into the system. And it's going to take resources. It's, and this is something that Pete was emphasizing as well. We have to maybe rethink a little bit what we use short-term funding for and long-term funding for. Maybe it's more important to have robots than it is to have lots of new research buildings. We'll have to make some of these trade-offs because there's a finite amount of resources available. But I think the opportunities here, I think especially for solving the basic science and mechanisms of human disease, are just tremendous. And it's engaging. And we do work with patient groups at the Chan Zuckerberg Initiative, as well as with basic scientists. We've been funding them. We're increasingly thinking about funding at that interface. It's so inspiring for a scientist to have a relationship with the patient group to make the work they're doing come to life. And, the sort of virtuous cycle of physicians, the scientists, the patients, as a way of uncovering mechanism and of moving, again, that key innovative step forward,
Right.
is something that could stand more attention, I think.
Terrific. Mark, the response to COVID has been interdisciplinary, intense, and effective, with some gaps, but mostly effective, at least around some aspects. Are there ways to think about an organization such as Stanford in terms of initiatives to build interdisciplinary activities that are very forward thinking, that are, instead of siloed academic disciplines, much more about the future of application of science with the cycle of learning that happens between the problem and the evolving science?
Absolutely, Tom. And in fact, it's a path that we've been on, and I know other universities have as well. And maybe I can make a few quick points on this. First, one of the things that we saw with COVID was, breaking down of the barriers. People were so motivated to focus on the disease. There's this rapid pivot among our faculty to focus on the disease. And by the way, we're talking about the biomedical aspect here, but this was true also of our social scientists who looked at the societal impacts, at our historians who looked at past pandemics to learn lessons from them. Everybody focused on COVID in order to think through how we could contribute to solving the problem. On the biomedical side, what was very inspiring was a lot of the obstacles that typically exist just melted away. People started collaborating with people at other institutions very rapidly. For example, Corey and I and the Chancellor of UCSF got together with our colleagues at the three institutions, CZI, Stanford, and UCSF, to put together a really important epidemiological study to study how the disease was progressing and try to derive knowledge that could be used to get kids back into school eventually. And we blasted through, all the issues around, agreements and intellectual property and the like. Our intellectual property offices partnered with those of other major institutions to come up with a standard way of doing things so that no intellectual property issues would get in the way of advancing solutions. There was a real premium on going quickly, not cutting corners scientifically, but getting rid of obstacles that you can understand why they're there, but they don't have to be there in that form. We do have to tend to those issues of intellectual property, but can we do it in a better way? Interactions with the regulators suddenly became much more rapid and systematic, and a lot of lessons have been learned also from that. I think there was huge progress that was made, and we know that even more can be made. So I'm hopeful that as we emerge from this as a society and as an ecosystem, that we preserve those gains and we don't revert to the silos that we had before. Within our institution, we had to attend to the following. There were so many people doing things. There was a lot of, there were missed opportunities for collaboration and coordination. So we did the simple thing of putting together a website with 400 faculty members entered their programs. They could look to see what other people were doing. So I think the spirit that came from that, I think, is something we really have to capture and bottle and deploy going forward. And the private sector, the not-for-profit sector, the academic sector, the regulatory and government sectors, all working hand in glove to go forward. We've, in terms of, your question was about, at Stanford, can we ensure more interdisciplinary work? And the university under my predecessor had been on a journey to break down the silos. And we found mechanisms to bring people together, often around projects where, by providing resources and providing infrastructure to enable them to do that, that you can get people together to focus in this way. This is something that you have to nurture every day. It's easy for people to revert. But there are now well-worn mechanisms. Again, we didn't even invent most of them. It's just a question of deploying them and applying them within your ecosystem to try to create that kind of collegiality, that interaction, and the cross-disciplinary work. It requires leadership. We have extraordinary leaders. Chetan Khoshla is heading our Innovative Medicines Accelerator that has this drug prototyping unit. Peter Kim, who is well known to all of you, has been playing a key role. Carolyn Bertozzi as well, Carla Schatz. So leadership within the university has taken on the mantle of fostering this kind of interdisciplinary research. That spirit really helped us during COVID, but it's something we saw across the country, across the world, and we really have to build on that.
I totally agree. Pete, I know that you have thought deeply about the direction in which the Institute will go, TSRI. Maybe you can just share some perspectives on, how the best of interdisciplinary science emerges from Scripps.
Yeah, it's an interesting question. How do you maintain a focus on basic research and technology development and also applied science, making drugs? They're very different cultures. And in fact, people who do applied science in nonprofit settings face career challenges, If you try and make a drug, if you fail, which is often the case, maybe you don't get publications, and that negatively impacts your academic career. So I think we need to, if we want to incentivize, kind of applied life science as well as basic science, we need to rethink how we reward people in the academic world. Also, in terms of education, we educate people to solve our problems as faculty members. We're pretty short-sighted in challenging students to what problems would you want to solve and should you solve when you're out of my lab, So I think the culture, the traditional academic culture has to break down of what we view as success. And I think how we educate our students needs to change from problem solving to the ability to identify interesting and new problems. Those are two important things that have to happen. And as I think about where Scripps is going to go, we need to continue to bridge these two worlds, because I think there's a huge opportunity there for academic institutions to have an impact. But we also need to change the way we think about success and nonprofit research institutes. I think my philosophy is we don't, except for this idea of doing outstanding basic research and technology and applying that to important medical problems, we tend not to be overly defined where we want to go. It's simply hire the best and brightest young people And if you hire the best and brightest young people and give them resources and nurture them, they'll decide the directions of science. And I think what we're seeing, and what both Corey and Mark commented on with COVID, is people really want nowadays to solve problems that impact society at all levels. And I think we're starting to see more and more of that in young people. And, you know, as you see young people focus on problems, a lot of those, the papers you're starting to see are making molecules that really impact human disease in interesting ways. So, you know, I think the young scientists are taking this in the right direction. We just need to create the infrastructure to make sure we don't get in their way, Tom.
Yeah, I certainly agree. We have a few minutes left. I would just ask each of you to reflect on, you know, a single pressure point that you might emphasize as the most, you know, potentially impactful, that from your seat you can change. If you got to just, you know, take the CZI initiative, or Mark Stanford or Pete Scripps, or what would you like to change in a sentence or two that would really impact the future of translation science?
So Tom, if I could just start, I would like to change just how we reward people. I mean, we need to encourage risk taking, okay? And if people work on high risk projects, be it a basic research project or translational research, if they fail, okay, that doesn't mean they're failures, okay? It means they may have picked a really hard problem and done the best job anybody could do. And we ought to keep those people and nurture those people. And so I don't think the current system really encourages risk taking, not only in the academic sector, but also certainly in the commercial sector.
Mark, I'm going to close with Corey. Yeah.
Well, maybe just a few thoughts. First, I think we have mechanisms now. I talked, you know, we heard about Caliber, you know, CZI, Stanford, mechanisms to assist the translational process as it starts in academia. We need to hone those mechanisms further where they already are. But I'd also like to see us make them scalable and transportable so that other institutions around the country can adopt them. We'll all be much better if it's being done not just, you know, in Cambridge and San Diego and the Bay Area, but across the country, you know, in all the great research institutions that we have, you know, in the Midwest and the South and the Northwest and so forth. So getting that to diffuse across the country, I think would be very important. Thinking about how we could encourage that, and it has to be, it can't be the results of specific individuals who happen to have the experience like Pete, who can do it in a bespoke manner at their institution. It has to be made accessible even in institutions where people might have less of a history of doing this.
And then Corey, if I may just ask you to close for us.
Yeah, I just think that we need to broaden the definitions of success. I believe that science is doing great, actually, and that it could do better by including more kinds of ways of being successful. You know, you're looking at a group of people who really have the ability to make these changes. Mark is the head of a great institution. One of the ways that you create incentives in science is through hiring people. And, you know, I'm leading a philanthropy, and one of the ways in which you make changes and create incentives is through funding people. And I'm proudest in some ways of some of the funding we've done that's been unconventional, like funding people who write open source software, and funding imaging scientists who work in imaging facilities at different locations, who are kind of technical experts and not necessarily faculty members. I think we can broaden the definition of success and, again, take into account that things don't work all the time. We will create a more creative workforce.
Well, that's a wonderful close. Thank you all. This was certainly for me very, very enjoyable conversation, and I'm grateful for each of your perspectives. Thanks.
Thanks a lot, Tom.
Thank you.